So let us learn about the rostrocaudal patterning of nervous system. This is the schematic representation of the nervous system during its earlier stage of development. We have the prosencephalon, mesencephalon, rhombencephalon, which then further gets organized into telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And then we have the caudal neural tube. We also have certain flexures like the cephalic flexure, pontine flexures, and cervical flexure. But then how does this diversity come about? For that, let's go back to the organizer. Initially, it was believed that this organizer was uniform in character and induced a uniformed neural plate. Further studies confirmed that the case in point was quite different, and it is in fact regionally specified and secrete factors involved in rostrocaudal patterning as soon as induction commences. So then we have established that the organizer is not uniform and is regionally specified, and that it secretes factors which are involved in rostrocaudal patterning. What are these factors? Now, one of them is WNT, which is an acronym based on the founding family members, that is Drosophila wingless protein and mammalian INT proto-oncogene protein, and other some proteins like retinoic acid and FGFs. Now, the crazy thing is that at the rostral level, the net level of WNT signaling activity is low, and it increases progressively caudally. And this is because that the mesodermal cells of the organizers, they secrete more WNT in the caudal region, and then there are certain factors which inhibit WNT signaling at the rostral region. Now, these results suggest that an anterior character is default state for neural tissue. So here, let me reiterate the point that when the ectodermal cell was isolated from its nearby cells, we saw that its default was the differentiation into neural tissues. And here we see that in the absence of any secreted proteins like WNT, the cells transform into an anterior character type of cell. So this suggests that an anterior character is default state for neural tissue. And with signals like WNT, they impose a posterior character. So now we have arrived at a point where we have established that the anterior character is the actual default of ectodermal cells. Let's dig in further and see how signals from within the neural tube patterns the forebrain, midbrain, and hindbrain. So we get the initial signals from mesoderm and endoderm, but now for the patterning of cells within the neural tube, like into forebrain, midbrain, and hindbrain, we have organizing centers within the neural tube, and one of them being the isthmic organizer, which forms the boundary between midbrain and hindbrain. So here we see that in the black shaded region, that is supposed to represent the isthmic organizer, and the green cells, they are the dopaminergic neurons of substantia nigra and ventral tegmental area, and the red cells, they represent the serotonergic neurons of red nucleus, so which lie just rostrally and just caudally to isthmic organizer respectively. At this point, I would like to introduce homeodomain transcription factors whose expression sort of lays out the rostrocaudal positional characters of the neural plate. What is homeodomain though? It is that section of protein that binds to specific DNA sequence in the regulatory regions of gene, changing the gene's transcription. It was found that in the forebrain and midbrain, the cells express OTX2, and in the hindbrain, the cells express GBX2, both of which are homeodomain transcription factors. The transition between these is located in the midbrain-hindbrain boundary, and this is that portion at which the isthmic organizer will emerge after the neural tube closure. At this midbrain-hindbrain boundary, other transcriptional factors are expressed, like EN1, which controls signaling factors WNT1 and FGF8. WNT1 is shown to maintain the expression of FGF8, and FGF8 spreads in both directions, that is towards the midbrain and towards the hindbrain, or rostrally and caudally. But the cool thing is that the same molecule, which spreads in both directions, lead to proliferation of different types of neurons. The reason is the presence of homeodomain transcription factors, ODX2 and GBX2, in these two different regions. Now, the interpretation of this is that a small number of secretory products like FGF, BMP, etc., produces variety of neurons due to the difference in the transcriptional domains, which allow cells to interpret their actions in different ways. To see other examples would be to look at anterior neural ridge, which secretes FGF, which goes on to pattern the telencephalon in its glorious complexity, very meticulous. And the zona limitans intrathalamica, which secretes sonic hedgehog protein, which patterns the nearby cells to give rise to the nuclei of the thalamus. Let's dig into the last segment of this video, that is the subdivision of the forebrain and hindbrain into segments or compartmental units that are arranged along the rostrocaudal axis. To illustrate the mechanism of segmentation, we will use the formation of rhombomeres 3 and 4. So in the forebrain, the segments are called prosomeres, and in the hindbrain, they are called rhombomeres. Rectified in the following way: CROX20 induces the production of EPHA4, and HOXB1 induces the production of ephrin B3, both of which are cell surface recognition and signaling molecules, and they transmit repulsive signals to each other, separating these cells. So they act via repression. To conclude, the inductive or adhesive interactions combine with repressive or inhibitory ones to pattern the nervous system. So this was what I wanted to talk about in the chapter of patterning of nervous system and the references principles of neuroscience, the 6th edition. I hope you guys enjoyed it, and thank you so much.